Confirm me, am I audible to you?
Recording in.
Confirm me, am I audible to you? Good morning. Good morning. Okay, let's start our discussion. Good morning. See, in yesterday's class, we have completed up to Coelenterata. Good morning. In today's class, we are going to see about the helminthes. In helminthes, there are two things. Okay, we have to study about two phylums under this helminthes. One is Platyhelminthes and another one is Ahelminthes. Okay, in today's class, we are going to see about Platyhelminthes and what are the parasites which are coming under the phylum Platyhelminthes. Clear? Okay, this is the general characteristic features of Platyhelminthes. So the first thing you have to keep in mind when it comes to the helminthes category is they are going to cause some of the diseases to human beings or any other host, but it is in which form? Parasitic form. Okay, either it is parasitic or commensal form. What is commensal? Commensalism. Commensalism is a symbiotic association between two organisms, which one of the organisms gains benefit from the host and other organisms neither benefited nor is going to getting harm, which means plus zero. Okay, one is getting benefited and another which is not going to getting benefited, either it is not going to get benefited or neither harm. Okay, fine. Tissue organ grade of organization. The body cells aggregate into definite tissue and tissue makes up the organ. So this platyhelminthes, it is from tissue to organ grade of organization. See, up to coelentrates, you have to understand what are all the difference between the coelentrate characteristics and the general characters of platyhelminthes also. Up to coelentrates, what we have studied, it is diploblastic in nature. But from Platyhelminthes, see this Platyhelminthes, it is also called as what worms? Flatworms, because it is flat in structure. Okay, flatworms, and it is triploblastic in nature. You have to understand from which phylum the triploblastic character have been emerged means you should know that is from the Platyhelminthes. So what is triploblastic? It is made up of three embryonic germ layers. What is that? Ectoderm, endoderm, and mesoderm. From ectoderm, some of the organisms is going to emerge, and from mesoderm, some of the organisms is going to emerge, and from endoderm, it gives rise to some of the organs. Clear? And it is bilaterally symmetrical. So up to coelenterate, what we have studied radially symmetry or the biradially symmetry. But from platyhelminthes, you should make the difference here. From platyhelminthes, it is shown here as bilateral symmetrical, which means we have to cut an individual at only one plate to get an similar equal halves. Okay, fine. Bilaterally symmetrical with definite polarity of anterior and posterior ends. What is anterior? Here refers to head region. Posterior refers to tail region. Okay. Now, dorso-ventrally flattened. As it is a dorso-ventrally flattened throughout the organisms which is placed under the platyhelminthes, we are saying this as a flatworms. Okay. Why is it so? Because it is dorso-ventrally flattened. Clear? Now, here usually with a well-defined ventral surface bearing mouth and the gonophore. And body here is unsegmented. You should note a difference between the platyhelminthes, nemathelminthes as well as the annelids. From annelids, we are going to study the body is segmented. But up to the nemathelminthes or the platyhelminthes, we are studying here is body is unsegmented in nature. This also makes a difference. Okay, here acoelomate, the most important characteristic features of the platyhelminthes, where the question have been frequently asked. See, it is dorso-ventrally flattened. It is also called as a flatworms, and also the platyhelminthes is acoelomate in nature, which means what? There is no body cavity seen here in the platyhelminthes. The spaces between various organisms filled with a special mesodermal tissue, the mesenchyme or the parenchyma. Okay, but in case of the coelomate animal, what will happen there? There is space between the outer ectoderm and the endoderm, and in between these two, the body cavity will going to arise. But here in acoelomate animal, the gap will be filled by what? Mesenchyme or the parenchyma or the special mesodermal tissue, whatever we can say them. And the most important thing, from where the because of which structure these kind of worms is going to attach with the host, means these adhesive structures like hooks, spines, suckers, and adhesive secretions are common in parasitic forms. So this is the major category for all the organisms who show their life cycle as a parasitic forms are these hooks, spines, and the suckers. Suckers is for the mainly for attachment. Spines is for the defense. Hooks again for the attachment. Okay. By this structure, these parasite is going to attach to with the host, whatever be the host it is. Now, epidermis, cellular or syncytial, frequently ciliated, absent in some. Which means what? Epidermis is not frequently seen in any of the parasitic organism. Now, this muscular system is mesodermal in origin. We have studied these platyhelminthes as what? Fibroblastic in nature. It has a third important layer between the ectoderm and endoderm is mesoderm, and the muscle which is going to originate from this mesoderm. Okay. Now again the question. Up to coelenterata, we have studied that what are all the structures which have been originating nerve structure and muscular system. But of course, that is not well differentiated. But where it have been well differentiated in the phylum Platyhelminthes. In muscular system, it is mesodermal in origin. Now, longitudinal, circular, oblique muscle layers beneath the epidermis. These are all an extra point. There is no point of remembering all these things. Just think muscular system is developed and that too from the mesodermal origin. Now, digestive system. Digestive system is branched and incomplete without anus. Why it is said to be as an incomplete? It has been start from the mouth, but it is not end with the anus. That is why the digestive system is incomplete. Okay. Now, altogether absent in Acela and Cistola. These are all the class of the platyhelminthes. We are going to study about that. Now, see, skeletal system, respiratory system, circulatory systems are wanting. What is the meaning of this point? What is the meaning of this point? Quick. Skeletal system. I'm going to say two statements. You are going to say yes or correct, yes or no. Okay. See, in platyhelminthes, in platyhelminthes, very good. Absent. See the skeletal system, yeah, very good. Not developed completely. Skeletal system, respiratory system, circulatory system, they are not present in the platyhelminthes. They are wanting. Wanting means in the absence of these organisms. Clear? Fine. And the most important points, excretory system. This excretory system includes the lateral canals and protonephridia. So you have to remember this point compulsory. See, this protonephridia is an excretory organ, and what this protonephridia consists of? Which type of special cells? That is called as the flame cells. In yesterday's class, I have explained while dealing with the MCQ. When you heard about the name platyhelminthes, you should know what is the other name of platyhelminthes or what is the common name of platyhelminthes and what is that? What is that? Flatworms. Okay, and flatworms, and this has then excretory organs which is present only in the phylum Platyhelminthes are flame cells. Okay, their structure is called as the protonephridia, and this protonephridia is made up of which cells? Flame cells. These are all the things you have to remember. Now see, nervous system is primitive, ladder-like. Okay, so you have to remember from platyhelminthes also, the nervous system is not completely developed. It is primitive in nature. That is ladder-like and comprises a pair anterior ganglia with the longitudinal nerve cords connected by the transverse nerves. These are all not necessary, but you should know nervous system is present in platyhelminthes, but primitive in nature. Sense organs are simple. Okay, and what is that sense organs? Eye spots or photoreceptors. Okay, this is the sense organs in the platyhelminthes and mostly monoecious in nature. Again, what is monoecious? Why I am asking repeatedly the same questions again and again means by repeatedly saying only then you can remember easily. Okay, what is monoecious? And what is hermaphrodite? Quick. We have discussed in the class repeatedly. So don't make a mistake. Just tell me what is monoecious. Again, you're getting confusing, I think. Not developer. Very good. Having male and female reproductive organ in the same individual. Yes. That is, yes, both sexes in the same individual. It is bisexual in nature because both sexes is just placed in the same individual. That is monoecious. So what we have to conclude here? This platyhelminthes or the flatworms or the monoecious organism. Okay, complete the complex reproductive system because both are present in the same individual, right? When well-developed gonads, gonads, accessory organs, eggs mostly devoid of yolk. See, eggs generally consist of what? Yolk, and yolk provides nutrition to those contents which is present inside the shell. But here, eggs is mostly devoid of the yolk. Okay, yolk produced separately in the yolk or vitelline glands, and where the yolk is present, the yolk is present in the vitelline glands. And fertilization is internal. See, you have to remember which are the phylum undergoes internal fertilization and which are the phylums which have external fertilization. Okay, so internal fertilization means what? The fertilization or the fusion of male and female gamete happening within the body of the female organism, and that fertilization may be cross on the self. And I hope you remember the difference between the cross fertilization and self fertilization. What is self fertilization? The fusion of male and female gamete occurs within the body of the organisms which is produced by the same individual. In case of the cross fertilization, male gamete is going to synthesize in one of the organism and the female gamete is synthesized in other organism, and these are going to get fused. Okay, and as a result of fertilization, zygote is formed. That is said to be as cross fertilization. But in case of platyhelminthes, both cross and self fertilization has been seen here. When the self fertilization is going to happen and when the cross fertilization is going to happen, that also I have explained you multiple times. If the male and female gamete produce at the same time, then which fertilization is going to happen? Self-fertilization. If the male gamete produced at one time and the female gamete is produced at some other time, then it is said to be then it is these organisms going to prefer the cross-fertilization where the egg is readily available. Okay. Now, development is direct or indirect. Development have been direct development or indirect development. Straight away the young adult gives rise, sorry, straight away the young or gives rise to an adult. That is said to be as direct development. Instead of giving rise to an adult from the young, they are undergoing some of the larval stages that are said to be as an indirect development. Okay, see, usually these development is indirect in the endoparasite. What is endoparasite? Generally, the parasite are of two types. One is ecto and endo. In case of endoparasite, the parasite have been living within the body of an host. That is endoparasite. If it is present outside the surface of the body of the host means, then it is said to be as the ectoparasite. Okay. In endoparasite, generally it is indirect development. Okay, that's all about the general characters of Platyhelminthes. Now we are going to see about the classification of Platyhelminthes. And the first class is Turbellaria. Okay, here mostly free living flatworms. Some are ectocommensals and endocommensals. Commensals means we know one of the organisms getting benefited and the other is neither getting benefited nor getting harmed. And ecto means living outside the body of the host. And endo means living inside the body of the host. Okay. Now, some are parasitic. Parasitic means what? One is getting benefited, other is getting harmed. You should know the symbols also. It is very easy for you to understand. Now, body is dorso-ventrally flattened. Because of this character, we are saying Platyhelminthes are the flatworms. Okay. Now, body is unsegmented. We know that only from Annelida, all the organisms possess the segments. Up to the Nemathelminthes, there is no structure called segmented or metasegmentation. The body is covered with a cellular epidermis with mucous cells and is partly ciliated. Okay, now adhesive organs abundantly present. Why? Because only because of this character they can act as a parasitic in nature. And what is their adhesive organs? Hooks and suckers here are absent. And what is going to present? We have studied hooks, suckers, and the spines. And here they are having spines because hooks and suckers are absent here. Here, what is the sense organs? Tangoreceptors and chemoreceptors. Only then it can sense the prey. Okay, and what is the sense organ seen in the double area? It is tangoreceptors and chemoreceptors. And digestive system consists of mouth, pharynx, intestine. Anus is not formed. And why? Because of this anus is not formed, we have said this digestive system in the phylum Platyhelminthes is incomplete. Okay, why? Because anus is absent here. And excretory system consists of protonephridia and the flame cells. This is the most remarkable point than any other point. Flame cells. Mostly hermaphrodite. We know that it is monoecious or bisexual and sexes are not separate. Both the sexes are present in the same individual. Reproduction is sexual, asexual and by regeneration. So we have to know both sexual, asexual or by regeneration. Everything is possible. At the end product of all these type is going to give rise to another individual. That's it. Only this, the mechanism, the mode of reproduction is different and life cycle is simple in nature. Those who are placed under the turbal area. And examples are very, very important. Planaria, bipalium, autoplana, notoplana. Okay, you have to remember what are all the examples. And the second class is Temnocephaloideae. As the name suggests, it has cilia in it and it is covered with a cuticle. The best examples, see, the best example for is Temnocephala. Okay, from the name you should know that. Rhabdites occurs in the ectodermal cells of the tentacles. And what is rhabdites? We are going to discuss in the upcoming slide. See, you have to remember this point compulsory. Rhabdites occur in the ectodermal cells of the tentacles. Here, gut is present. The largest here there is a large posterior sucker. Here, spines is present because hooks and suckers are absent. But here, what is the adhesive organ present in Temnocephaloideae? Is sucker. Okay. Turbal area example is planaria, bipalium, autoplana, and notoplana. See, planaria, bipalium, autoplana, and notoplana. These are all the examples of turbal area. Clear? And see, protoglottids are never formed. See, the protoglottids are very important structure when it comes to the platyhelminthes. Okay. See, proglottids are never formed. Okay, these are all helpful in the reproduction part. And members are epizoic on invertebrates, chiefly freshwater crustaceae. They are found in some of the other organisms also, especially crustaceans. Members can be recognized by the possession of tentacles anterior to the mouth. How they can be identified? By presence of the tentacle which is present anterior to the mouth. And the best example is Temnocephala. Clear? Now, the third class is Trematoda. This Trematoda can be either ectoparasitic or endoparasitic in nature. This is commonly said to be as flukes. For example, liver fluke. Everything. The name which is comes with the fluke. Everything will be placed under the Trematoda. Okay. Now, the shape of the body is leaf-like, which is dorso-ventrally flattened. Epidermis and cilia are absent here in the body wall. Again, the body is unsegmented. It is covered with a cuticle. Here, the hooks and suckers are present. Okay, this is an important point. Here, the digestive system, again, it is incomplete because anus is not present, but mouth, pharynx, and the gut is present here. And the excretory system is again protonephridae and the flame cells. And hermaphrodite monoecious. Here, the ovary is single in number and the testes are many. Okay, life history is simple or it can be complicated, whatever it is. And the best example is fasciola hepatica. This can be asked in the question, what is the common name of fasciola hepatica? Or what is the scientific name of liver fluke? Whatever be the question is, and you have to know liver fluke means fasciola hepatica and it comes under the phylum Platyhelminthes. Diplozoans. These two are the best examples for the Trematoda. And the fourth class is Cestoda. See, this Trematoda and Cestoda is very important because they have an important examples of Fasciola hepatica and the Tinea species we are going to discuss in the upcoming slides. They are parasitic in nature, particularly endoparasite, because it is present within the host, particularly the intestine. They are said to be as the tapeworms. Okay, what is their common name? They are said to be as the tapeworms, and Trematoda is said to be as the flukes. And body wall does not have epidermis and cilia, but covered with a cuticle. Body is segmented. These segments are said to be as the proglottids, and it is rarely undivided. See, the anterior end is modified as a head, which is called as scolex. These are all the important structures. What is meant by scolex? The anterior end is modified as head. So, in tapeworm, the scolex is nothing but a head. And it is the head, which is nothing but a scolex, which is provided with an adhesive structures. And what is an adhesive organs present in the cestoda is hooks and the suckers. And the cestoda do not have the mouth and the digestive tract. This is also an important point. Up to the trematoda, what we have studied, anus is not present, but here they do not have mouth as well as the digestive tract. And the excretory system is again protonephridia. Again, the flame bulbs, which has the flame cells. Nervous system is consists of the pair of ganglia and the two lateral longitudinal nerve cords. It is not a well-developed nervous system, but it is also a primitive nervous system. And each segment is nothing but proglottids, contains one or two sets of incomplete hemiprodect reproductive system. See, you have to remember here each proglottid or the each segment contain one or two sets of complete hermaphrodite reproductive system. So all the segment which is consisting of the gonads. Okay, now the life cycle is complicated, usually two or more hosts. Okay, two or more hosts. The best example for this cestoda is Tinea species, Convoluta, and Echinobothrium. Okay, examples are important. Don't forget to read this. So don't, yeah. Okay, that's all about the characteristics and classification. Okay, any doubts with respect to this too? Clear, everyone? Shall we move to the next part? Good. Okay. So we have completed the general characters and classification. So they have in their syllabus, they have given some of the parasitic organisms. And what is the first thing? Tinea solium, Schistosoma species. We are going to deal with these two in today's class. If time permits, we will go further. Okay, the first parasitic organisms we are going to see is Tinea solium. Okay, Tinea solium. So straight away in your question, in your examination, they will raise a question. And what is that? What is the common name of Tinea solium? So take down in your notebook. The first point you should remember when it comes to Tinea solium is the common name for Tinea solium is the dwarf tapeworm. Yeah, the first point. Now, coming to the systematic position, the phylum Platyhelminthes, Cystoda. Just now we have studied what are all the characteristics of Cystoda and Eucystoda, Tanoidae, Tenidae, Tinea, Solium. Tinea is a genus and Solium is the species name. But when it comes to the question, if they gives in the option, Tinea is said to be as the genus, Solium, Tinea, Solium. So what is the species name here? In your exam, they are asking what is the species name, which option, which is the correct option, which refers to the species. And what will be your answer? This is the option 1, 2, and 3. Option 1 is Tinea, option 2 is Solium, option 3 is Tinea Solium. This is what everyone will made a mistake. See, Tinea is a genus, no doubt in that. It is a genus. Okay, and Taenia solium will be the correct species name. Solium is not a species name. When it comes to the question, you should very good. You should know that Taenia solium is the species name. But when they ask about the genus, what is the genus present in here means Taenia is the genus. Okay, but in your question, when they ask like this means you should know Taenia solium is the species. Clear? Okay, fine. Next, this is the life cycle of the Taenia solium. We are going to study about this and later we'll discuss this. Only then you can understand clearly. First thing you should remember is life history and the development. Okay. First one is copulation and the fertilization. The life cycle of Taenia solium is digenetic in nature. What is digenetic? I have explained when I talked about the protozoan diseases. It is digenetic in nature. Why it is said to be as digenetic in nature? Because very good. It completes their life cycle by involving into two hosts. Okay, it completes their life cycle with the help of two hosts. Only then it is said to be as a digenetic. In case if it requires only one host means then we can call them as a monogenetic in nature. So the second point you have to remember, very good. So the second point you have to remember when it comes to Taenia solium is it is digenetic. Okay, so take down. But the life cycle of tapeworm is simpler and without a free larval stage. Okay, without a larval stage. So the development is direct development. But some of the cases they also undergo a larval stages. Yeah. Presence of single tapeworm in a host diminishes possibility of the cross fertilization. So it can prefers the cross fertilization. The fertilization is preceded by the copulation. So only after the fusion of a male and female gamete, fertilization will be occurred, and which is accomplished by the insertion of cirrus into the vagina of the same or other proglotid to release the spermatozoa. See, insertion of cirrus into the vagina means cirrus is a part through which it is required. It is the structure which is required for insertion of sperm into the vagina. Okay, of the same or the other proglotid. See, I have mentioned you these are all the see. This is each it consider this as a segment like, but it's not an actual segment. Okay, so each segment, each proglotid is having a two male and female reproductive organ. So consider this as a male, this female. This is a male, this is a female. Okay. See now this male has to reach the female to fertilize. But what happens here? The male can also reach this female reproductive organ which is present in the same proglotid, or this male reproductive organ can also reach the other female reproductive organ which is present in the next proglotid or the segment. Okay, wherever the egg is readily available, the cirrus will reach the vagina to reach the spermatozoa or the sperm. Clear? Now it becomes possible when the common gonophores of two mature proglotid come in contact due to the folding of strobila. Okay, how it will be happen, how the male reproductive organ, yes, how it is going to happen means the male, yeah, it is a internal fertilization, because they both which is present inside they are going to fertilize within the body of an organism. See, the male here which is present, they reaches the other female reproductive organ or the female gamete which is present in the other segment by due to the folding of the strobila. Okay, this you have to remember. So the next thing, an anterior mature proglotids means it is a segment. Progyno, we have studied yesterday. There are two conditions. If the male reproductive gamete, which is present, is produced before the female gamete, means that is a different condition. And if the female, yeah, that is the exact reason. If the female gamete is produced before the male, that is progyno. Okay. And fertilization following copulation between two different proglottids is sometimes termed as a cross fertilization. When the cross fertilization will occur, if the male and female gamete are produced at a different time, the organisms or the parasite is going to prefer the cross fertilization to distinguish in form that occurring between the gametes of the same proglottid, that is self fertilization. Spermatozoa injected into the vagina. Now the sperm have been injected into the vagina. Now what is going to happening here? Swim down to the seminal receptacle, where this is the area where the ova have been stored and released by the ovary. Only then the fertilization is going to occur. The fertilization happening in the fertilization duct, where the vagina and the sperm is going to, sperm and the ovum has going to meet. Now fertilization is going to take place. As a result of fertilization, male and female gamete is getting fused, and what is the structure is formed? Zygote is formed. That's all about the fertilization. And after fertilization, it has to be protected. And what structure going to protect the xylem means? The capsule. Capsule is a structure going to protect the zygote. Zygote is also said to be as a egg cells, which is passed into the ootype, where each becomes associated with a large yolk cell or the vitelline cell provided by the vitelline man. So the first purpose of the capsule is to protect the zygote or the egg cells. The two, see, when it comes to the parasitic forms, there are many things I have mentioned, but I have to suggest you is what are all the things I am saying it is important. You have to read that. That is okay. That is sufficient. So what is the first point you have to remember is it is the common name, and second one is it is diuretic in nature. And we are going to discuss what are all the two hosts they are going to prefer. And first point is how their life history is. They are going to fertilize either by self fertilization or the cross fertilization. And the second thing is, as a result of fertilization, zygote has been formed, and what that zygote or the egg cells is going to protected by the capsule formation. Okay, no. The two become enclosed in a thin shell or the chorionic membrane, formed by the material excluded by the yolk cell. See how the capsule is going to form. Anyhow, the motto of the capsule is going to protect the is the zygote, protection of the zygotes or the egg cells. How this is going to form is by the membrane, which is by the material which is produced by the yolk cell. Okay, now the capsule so formed passed into the uterus. Now it has been the zygote have been encircled by the capsule. Now it is passed into the uterus. In the place of uterus, the further modification of the egg is going to take place. Now, passage of the capsule into the uterus, it is lubricated by the secretion from the Meigs gland. So it is present in the female reproductive organ. You should know that. And what is the purpose of Meigs gland means? See, initially it is present in the fertilization duct, and it has to reach the uterus, and it has to reach the uterus. To move from this place to this place, it has to move very smoothly. For that, it has been secreted by some of the mucus so that it can pass very easily. And what is the structure which is going to secrete this lubrication is Meigs glands. Now, as more and more capsule pass into the uterus, it develops into the lateral branch to accommodate them. Now, oncosphere formation, the most important point, oncosphere formation. And in this oncosphere formation, what are all the things going to happen? Means cleavage and morula formation. Cleavage, morula formation, and hexaganth and oncosphere. And what is cleavage? After the egg fertilization is going to happen, and after fertilization, zygote is formed. After fertilization, the zygote is covered by the capsule, and in that capsule, it is going to divide. Okay, only then the three different germ layers is going to give rise to some of the organs. And zygote or egg cell undergoes cleavage when the capsule is in uterus. Okay, when it is undergoing the cleavage, when it reaches the uterus. And what is the type of cleavage is seen in here means holoblastic cleavage. You can read this in the developmental biology, but here just know this holoblastic cleavage means the entire zygote will undergo a complete cleavage like this. complete cleavage, and it is unequal. It is not an equal, it is unequal. The first unequal division results in a large megamere and a small embryonic cell. See, this is said to be as the egg. Okay, this is said to be as the yolk, and this eggcell or the zygote, it is covered by the chorionic membrane, eggshell or the capsule, and this has been protected by the zygote has been protected by the eggshell, and it is going to nourish by the yolk cell. Okay, yolk cell is the one going to give nourishment, and the first cleavage gives rise to the megamere, which is larger in nature, and the smaller embryonic cell. This embryonic cell is the smaller. See, megamere and the embryonic cell. So the first unequal division gives the larger megamere and the smaller embryonic cell. Okay. Now, after this, morula. This megamere, see, first unequal division gives rise to what? Megamere and embryonic cell. Megamere is larger in nature, and embryonic cell is smaller in nature. Now, what is the structure which is called as the stage of morula is going to form, and how it is going to form? This megamere divides further and forms several similar megameres, while embryonic cell divides repeatedly, producing two types of embryonic cells, larger mesomeres and the smaller micromeres. See, let me explain you completely. Initially, it was a zygote, and as a result of cleavage, it gives to first unequal division gives rise to the megamere and the embryonic cell, and this is larger and this is smaller, and this larger megameres gives rise to the further megameres, many megameres. And in case of embryonic cell, for each division it gives rise to the two different types of cells. One is larger mesomeres and the smaller micromeres. Okay, mesomeres and micromeres, and this larger mesomeres is little bit smaller than the megameres. That is why it is said to be as the mesomeres. It is micromeres, it is macromeres, and this is mesomeres. Clear? Now, the second one, if you clear with this point, only then you can understand the next point. So if you have any doubts, just ask me right now. Okay, the three types of cells. What is the three types of cells? This megameres, larger mesomeres, and the smaller micromeres. These type of cells result from the zygote. We know that. And smaller micromeres, medium mesomeres, and the larger megameres. These are all the cells. And micromeres, that is the smaller micromeres, forms a rounded mass. This is the structure we can call it as a morula. It is surrounded by an inner envelope which is said to be as a mesomeres. Clear? See, this is the micromeres. This is the micromeres. It is surrounded by the mesomeres as well as the megameres. Okay, it is surrounded by the inner envelope of mesomeres and outer envelope of megameres. Okay, now the yolk or the vitelline cell transfers its yolk to the megameres. See now, what is this? Is the structure? This is the structure. This is said to be as the shell. This is the shell, and this is the yolk cell. This is the yolk cell. This gives a nourishment to these micromeres, mesomeres, and megameres. And what is the function of yolk or the vitelline cell? They transfers its yolk to the megameres. Okay, they are going to give the yolk to the megameres. And after donating all the yolk content to the megameres, these yolk or the vitelline cell going to disappears. Now, the larger yolk megameres, because the yolk cell which contains all the yolk, it is transferred to the megameres. So now we are calling it as the large yolk megameres, fused to form an outer syncytial nutritive envelope. So this is transformed as it has the yolk, it forms as a nutritive envelope or outer embryonic membrane. So we have to know that this megamere is the one later develop a structure as the embryonic membrane. Now, this embryonic membrane nourishes the embryonic cells. That is nothing but the mesomeres and micromeres. And after donating all the nourishment to those mesomeres and micromeres, finally, it is also going to disappear. Don't worry, I'll explain you once again. Now, medium mesomeres form a thick. Now, all the nourishment have been received by whom? Mesomeres and micromeres. Now, this medium in sized mesomeres form a thick, hard, cuticalized and radially striated shell, which is called as the embryophore or the inner embryonic membrane. So, you have to remember this large yolk megameres transferred their structure as the embryonic membrane. This medium mesomeres is giving rise to or developed into the embryophore or the inner embryonic membrane, which is the one surrounding the morula. What is morula? Morula is nothing but the micromeres. Micromeres is said to be as the morula. Now, beneath the embryophore, which is nothing but what? Medium mesomeres is the one giving rise to the embryophore. And beneath the embryophore means below the embryophore, there is a thin basement membrane which is located. Okay, see the structure. This is the morula. Initially, what we can call it as micromeres. Micromeres is the structure. We can call them as a morula. And after the morula, we can see this is the megameres. This is an embryophore secreting mesomeres, which means what? This is the structure we are calling as the medium mesospheres. Later, it gives rise to the embryophore. Okay, now this is said to be as a megameres because all the yolk content which is present in the yolk cell, which has been transferred to these megameres, it becomes the yolky megamere, or we can call this the embryonic membrane. Okay. Okay, let me explain you once again. These are all that much important, but you should know the basic things. What is megameres, what is mesomeres and all. See, initially, what is happening? The first cleavage division. See, first the fertilization is going to happen. Fertilization. And after the fertilization, what is going to happen? Zygote is formed and zygote is protected by the capsule. And as a result of capsulated zygote, cleavage is going to occur. Where this cleavage is going to happening? In the uterus. In the uterus, and the first cleavage gives rise to and that cleavage is what? Holoblastic and unequal cleavage. And this cleavage gives rise to megameres and the embryonic cell. Okay, this megameres is larger and this embryonic cell is smaller in nature. Okay, up to this, I hope everyone will be clear. Now, this megamere gives rise to, and further the cleavage is going to happening. This megameres further gives rise to many megameres. Okay, gives rise to many megameres. But this embryonic cell further, for each division, they are further gives rise to two types of cells. One is mesomeres. Mesomeres and the micromeres. This mesomeres is the larger when compared to the micro. When compared to these meso and micro, megameres will be larger. So this is considered as the larger and this is the middle in structure, medium in structure, and this micro is smaller in structure. So we can call this embryonic cell is the one gives rise to one larger mesomere and one smaller micromeres. Okay, now there are how many types of cells have been formed? Megameres, mesomeres and micromeres. Three types of cells have been formed as a result of zygotic cleavage. Now, this micromeres is the one we can call them as a morula. A group of micromeres we can call them as the morula. Okay, don't get confused. This is said to be as a morula. And now, this morula is the one surrounded by the mesomeres. A group of morula is surrounded by the mesomeres internally. And externally, this morula is covered by the megameres. So you have to remember this mesomeres are, these morula is surrounded internally by the mesomeres and externally by the megameres. Anyhow, these two different types of cells going to surround the micromeres, which is called as the morula. Okay, now. And apart from these three structures, another structure is also present there. And what is that? A yolk cell. This is the one going to provide the nutrition for these three cells. Now, all the yolk content which is present inside the yolk cell, which is transferred to do what? These megameres. Okay, this yolk content will transfer to the megameres. What is that? All the yolk content which is present inside the yolk cell will transfer to the megameres. Okay. Now this after receiving all these yolk content, this yolk cell will getting degenerated because the purpose of yolk cell is lost. All the yolk content have been transferred to megameres, so no use of keeping their yolk cell is possible. So yolk cell is going to disappear. So after receiving the megameres, we can call them as the large yolk megameres. Large yolk megameres, and later this large yolk megameres is the one develop as an embryonic membrane. They develop as that what? Embryonic membrane. And this embryonic membrane, as it has yolk in it, this is the one provides nourishment to other two different types of cells. And what is the different types of cells? Mesomeres and the micromeres. Mesomeres and the micromeres. Clear? Now, and after providing the nourishment to these mesomeres and micromeres, this embryonic membrane or the megameres, again they are going to disappear. Now, after receiving all the nutrition from the embryonic membrane or the megameres, these mesomeres form as a thick, hard, cuticalized and radially striated shell, which is called as the embryophore. So you have to make a note of it. This megameres is the one called as the embryonic membrane. This mesomeres is the one going to called as the embryophore or the inner embryonic membrane. Finally, this is the one called the mesomere or the embryophore going to surround this micromeres. We can call them as a morula. And below these embryophore, we can call them, there is a presence of basement membrane. So you have to remember, between the mesomeres and the micromeres, there is a membrane which is called as the basement membrane. That's all about the morula. Any doubts with respect to this? If you have any doubt, means you can ask me now. If not, we'll move on to next point. Hexagon and oncophore. Now, another structure which is going to produce, that is hexacanth and the oncosphere. Morula at its morphologically posterior end develops their three pairs of chitinous hooks secreted by differentiated cells, which is called as the oncoblast. See, just up to this you have to be very clear. Only then this slide will getting you very clear. Okay. See, morula is nothing but what? Micromeres. Micromeres, which is said to be as the morula. Now, in its posterior end, there is a development of three pairs of chitinous hooks. Three pairs, which means how many hooks? 6 hooks. See, 3 pairs means each contains 2. So, how many hooks present? 6 hooks present. This is also important point. Here one pair, and here one pair. So totally 3 pairs of chitinous hooks secreted by, and here comes the question. These hooks are going to secreted by which types of cells? Oncoblast. Name of the cell is oncoblast. Now this 6 hooked embryo. See, as I said, 3 pairs, so 6 hooks. This 6 hooked embryo, it is called as the hexacanth. Okay, and what is hexacanth means? You should know it has this six hooks. Only then it has been called as the hexacanth. And these hooks have been synthesized by which type of cells? Oncoblast. And another thing, what apart from these hooks, what are all the things have present in the hexacanth means penetration glands. This penetration glands is very important for the penetration of something, whatever it may be. It is surrounded by the 2 hexacanth membrane. So you have to know this hexacanth, it is surrounded by the 2 different membranes, that is called as the hexacanth membranes. So together with the hexacanth membrane, this hexacanth membrane and the hexacanth, this is said to be as the oncosphere. So what is mean by oncosphere means, see, initially the hooks is present. Okay, 3 pairs or 6 hooks is present, and with this hooks is created by the cells called the oncoblast, and we can call them as the hexacanth. Okay, we can call them as the hexacanth. Now this hexacanth is surrounded by the 2 different layers, and with this, and these 2 different layers is said to be as the hexacanth membrane. And besides this hexacanth membrane, this hexacanth, we can call them as the oncosphere. Okay. Penetration glands is going to Penetration glands is going to penetrate something, whatever it may be. Okay, via this gland, everything is going to penetrate, whatever it may be, based on their purpose. Clear? Now, penetration glands, I'm going to explain. Mainly, it is for reproduction. Okay, for penetration glands means it is going to helpful in the penetration of sperm or ovum, whatever it is. Not only that, apart from that, many other thing also happening in the penetration glands. Okay, it loses the original thin shell or the chorionic membrane so that embryophore forms its outermost covering. See, look at the structure. See here itself. Look at the structure. This is the hoof and this is the hexagon, and the hexagon is covered by the two different membrane which is called as the hexagon membrane. So this we can call them as the oncospheres. Okay, now it loses the original thin shell. This is the thin shell, outer thin shell. We can call them as a capsule. This will be lost here, and after that it looks like this. Now, so that the embryo for after the shell become lost, now which is act as the outer most covered means this embryophore. Initially, what we can call this embryophore as medium mesomeres. Remember, this medium mesomeres is the one going to convert the structure as their embryophore. And once the eggshell will be lost or the chorionic membrane will be lost, which will act as the outermost covering, obviously the embryophore. By the time oncospheres are formed, the proglottid becomes gravid and increases in size. Further maturation will be happening here. See, once the oncosphere will be formed, which is the hexagon will be surrounded by the hexagon membrane. That is said to be the oncosphere. Once it is formed, the proglottid becomes gravid. Nothing but the segments. The gravid, and this will be increase in their size. Okay, and these are all the increase in their size. This is not necessary. For that, for a sake of information, I'll explain. In uterus forms 7 to 13 lateral branches on each side containing this much of oncospheres. In Tinea saginata, another intestinal tapeworm of man. This is another intestinal tapeworm. Not only Tinea solium, Tinea saginata is also a parasitic tapeworm. Uterus of gravid proglottid has 16 to 20 lateral branches on each side. Leaving the highly branched uterus, the remaining structures of reproductive system will degenerate. These are not that much important. Now it is completely formed with its structure. Clear? Now it has to complete their life cycle. For that, it needs the host. And what is C in case, as I said, Taenia solium is what? Digenetic in nature. Digenetic means it needs two hosts for their completion of life cycle. So the first host it is going to introduce is pig, but it is a secondary host. Why it is called as a secondary host? Because the sexual reproduction or the sexual maturation will be happening only in the primary host. First host or the secondary host it will be pig. The second host, but the primary host is the second host, that is humans. This will be the question. Say, this will not be asked in the question, this will not be asked in the question, this will not be asked in the question. But what will they ask? This digenetic. What are all the two different types of hosts seen in the Taenia solium? This will be frequently asked. What is the host? Pig and human. What is the secondary host? Is said to be as pig. Primary host is said to be as the human. Okay, where the sexual maturation is going to happen only in the human. That is why that is said to be as the primary host. Now, how they have been infecting the pig? This will be the question here. See, gravid proglottids at the posterior end of strobila detach in group of 4 or 5 and pass out with the host feces. Now, on ground, the proglottid eventually disintegrate, setting free thousands of oncospheres. So, oncosphere, it is releasing in the fecal matter. The secondary or the intermediate host acquires the infection by ingesting the oncospheres. See, consider this a pig. You know that how the pig will be? What is the feeding mechanism. What is the feed for the pig? Generally the fecal matter of the human, and the human consists of lots of oncospheres in it. The human fecal matter consists of lot of what? Oncospheres. This proglottids contains lot thousands of oncospheres. After degeneration of this proglottids, they setting free thousands of oncospheres, and these oncospheres will present in the fecal matter of the humans, and that can be consumed by the pig. Now the pig will ingest the oncospheres and acquires what? Infection. Lots of oncosphere will getting into the system of pig. Now pig which regularly feeds on human excreta is usually the secondary host. Pigs which is regularly eating the human excretory matter, that is usually the secondary host. But dog, monkey, sheep are also get the infection. But man himself may serve as a secondary host by ingesting the oncosphere with adequately cooked or the raw vegetables. How the man will getting the infection means by eating the pig. By eating the fecal matter of the humans, the pig is getting infection. And the pig, how the man is getting infection means by eating the pig. That is why eating those things are very harmful. You have to take care when eating those things, whether it is cooked, whether it is completely cooked or not. If it is inadequately cooked or the raw vegetables, there is we are going to vulnerable for those infection. Now auto-infection may take place in person who already serving as a primary host. Who is the primary host? A human is the primary host. Auto-infection is also there. If the parasite find a favorable condition against the man, is vulnerable for the infection. Okay, now that's all is important. This happens when due to the reverse peristalsis, the detached proglottids are carried to the stomach where the oncospheres are liberated. See, once again, the man himself, this is called as the relapses. The man also act as a secondary host. Again, they themselves act as a secondary host. Because of this peristaltic movement, once again they are going to reach the stomach area. Again, their oncosphere will be liberated, and from the oncosphere, again the life cycle is going to start. So just remember, the secondary host will be the pig, and the primary host will be the man. Migration within the secondary host. Now the pig has been the pig ingested the fecal matter. By ingesting those things, the infection will be, the pig will be getting infected. Okay, now the pig will be getting infected. Now what is the parasite is going to within the body of the pig? In the stomach of the pig, that is secondary host, the oncosphere loses its embryophore. What is embryophore? Medium mesoneer is the one giving rise to the embryophore, and the basement membrane by the action of acidic juices. See, in the secondary host, this oncosphere loses its the embryophore and the basement membrane. How this basement membrane and embryophore will be destroyed means by the acidic reaction. That is particularly the name of the protein is pepsin. The pepsin will digest these two things. Now, and after that, what will be present here is hexagon will be present. This embryophore and the basement membrane will cover the hexagon, and after destroying this embryophore and the basement membrane, what is will be released? Hexagon will be setting free, and then passes into the small intestine. And after that, where two persisting hexagon membranes, we know that hexagon will be surrounded by the two membrane, that is hexagon membrane. Only then it is said to be the oncosphere, and again it is covered by the embryophore and the basement membrane. These two will be lost, and this will be setting free. And after this, the hexagon membrane will also be lost by the alkaline juices. See, embryophore and basement membrane will be lost by the pepsin, that is acidic juices, and the hexacanth membrane will be lost in the small intestine. See, in the stomach, it will be lost. What is lost in the stomach? Embryophore and the basement membrane by the action of pepsin acidic juices. After the hexacanth will reach the small intestine. There, the hexacanth membrane will be lost by the alkaline juices. Now, only hexacanth will be spines will be present, hooks will be present. Hexagon now activated by the presence of bile salts. bores the way into the intestinal epithelium to reach the submucosal lymph or the lymph vessel. They are going to reach the lymph vessel. This is accomplished perhaps jointly by the six hooks and the penetration glands. Now you are going to understand what is the purpose of penetration glands here. To move from one place to another, these hooks and the penetration glands will help the hexagon to move from the small intestine to the lymph vessels. Now clear? Who asked this question? Lalita, right? Penetration glands is the one going to help the hexagon that will move from the small intestine to reach the lymph vessel. Okay, to move from one place to another, this penetration glands will be helpful. And with also the six hooks, these hooks, what is the purpose of hooks means? This is for attachment with the surface or the intestine, and by attaching with the help of these hooks and the mucus which is secreted by the penetration glands, with the help of these two, this hexagon move from intestine to the lymph vessel. Now, hooks merely anchor the hexagon to the intestinal wall. The secretions of the penetration glands dissolves the intestinal tissues. This process takes only 10 minutes. Now the hooks have no use and they shed off. Clear? Now the submucosal blood vessel carries hexagon to liver via the hepatic portal vein. See the submucosal blood vessel. Now from the intestine, the hexagon will reach the lymphatic vessel or the blood vessel. Okay, lymphatic vessel or the blood vessel. Now now the hexagon will carry to the liver. Now the hexagon will carry to the liver via what? Hepatic portal vein. Okay, hepatic portal vein. From liver, again they are going to reach the heart, and from the heart through the blood they are going to circulate throughout the body. It finally reaches the straped muscles, usually of tongue, shoulder, neck, thigh, heart, where it settles to develop into a bladder worm or cysticercus. This is the most important questionable area. This is the question, don't forget to remember this. See, in finally from the heart, I'm explaining you once again. Clear? See, the oncosphere will reach the stomach. Initially, the oncosphere will reach the stomach. In stomach, what will be lost? The embryophore. And what is the structure will be lost? Embryophore and the basement membrane. These two will be lost in the stomach due to the secretion of acidic juices, particularly the protein pepsin. Now, the hexagon together with the hexagon membrane will reach the small intestine. In small intestine, with the action of the alkaline juices, the hexagon membrane will be lost. Hexagon membrane will be lost. Now, from the small intestine to reach the blood vessel, what is going to happening here? Now, the after the hexagon membrane will be lost, only hexagon with the six hooks will be present. That structure have to reach the blood vessel or the lymphatic vessel. For that, which structure is going to help the hexagon? Penetration glands as well as the hooks. With the help of these two structures, it reaches the blood vessel and the lymphatic vessel. And after reaching the blood vessel and the lymphatic vessel, the purpose of hooks will be lost and the hooks will be destroyed. And from the blood vessel, it has to reach the liver. Okay, from the blood vessel, it has to reach the liver via the hepatic portal vein. What is it? Hepatic portal vein. From the liver, it reaches the heart. From the heart, it is going to circulate through the entire body. And after that, it reaches the striped muscle, which is present in the tongue, shoulder, neck, and thigh, heart, wherever it is. It is preferred to live in the muscle area where the muscle is large. Now, where it is settles to develop into a bladder worm. In these striped muscles area, where it is tongue, shoulder, neck, thigh, heart, they are going to form the bladder worm. Bladder worm, it is also called as a cysticercus. So the question, how the question will be asked means, bladder worm is the life cycle, is involved in the life cycle of which of the following parasite? And you have to know this is the life cycle. This is the structure developed in the life cycle of the Taenia solium, which is placed under the phylum Platyhelminthes. These are all the things you have to remember. And one more thing is, bladder worm, it is also called as a cysticercus. That is also the point you have to remember. Okay, now, however, these may develop into other organs such as lungs, livers, kidney, or brain. So all the organs are vulnerable to this parasite. Non-muscular vital organs, eye, brain, liver, or frequently become the sites of cysticercus formation. Cysticercus formation, nothing but bladder worm formation. Okay, now cysticercus or bladder worm formation. Now, as I said before, when reach from the blood vessels to the liver, it has been lost its hooks. And how they are transferred from this blood vessel to liver means via the hepatic portal vein. Now the hexagon is devoid of hooks, right? Devoid of hooks absorbs nourishment from the host tissue. Okay, where they are going to feed, they takes their nourishment from the host. Now, a central cavity appears as the cells in that region break down. It enlarges and becomes filled with the fluid consisting mainly of the blood plasma of the host, because that is the main source of the food for the cystic circus or the bladder worm. In that cavity, what is going to filled with? It is filled with the blood plasma of the host. Now, this fluid filled vesicle or the bladder, now it is called as a thin wall, consisting of an outer layer of thick syncytial protoplasmic mass and the inner mesenchymal or the germinal layer. You have to remember how the germinal layers or the three germ layers will be developed here. Now, at a point morphologically, the anterior, the wall thickens and the invaginates. See, this is called as the invagination. The invagination, which looks like a halo knob, differentiates into inverted scolex possessing the suckers, hooks, and rostellum. Again, the structure will be reappear. It is called as the proscolus. The embryo at this stage, it is called as the bladder worm. This is the structure looks like. Clear? In Taenia solium, bladder worm is of cysticercus type, which is characterized by the large vesicle and the one scolex. What is the characteristic features of cysticercus, which is present in the Taenia solium, is a large vesicle and one scolex. This can also be asked in the question. Now, that is why this bladder worm is referred to as the cysticercus. The wall, it is made up of the cellulose. So, as it is made up of a cellulose wall, it is also called as the cysticercus or the cellulosa. Now, the formation of cysticercus is completed in about 10 weeks in a pig. Cysticercus develop in an adult tapeworm only when ingested by the human host. This is also an important point. This cysticercus, to complete their life cycle, or it has to develop into an adult tapeworm, only it finds the human host. That is why Taenia solium is said to be as digenetic in nature. Only then it has been developed into an adult form. Up to then it is remains as it is. That is cysticercus. After certain point of time, it will be degenerated. In pig's body, it leads to quite an inactive life. See, cysticercus lead an inactive life. Just to keep them alive, it use their intermediate host. But to transform into an adult type worm, it has to find the human host and remains viable. See, of course, it is inactive. It can remain viable for the several years, which dies becomes calcified. For containing viable cysticercus, it is called as the measly poke for spotted appearance. This is not important, but it contains approximately 500 or more cysticercus in the 1 kg of pork. See now how the infection to humans will occur. Now the primary host will be man. By eating the undercooked measly pork, the man getting infection. Now the cysticercus, which becomes inactive in the pig, becomes active once it finds the primary host, that is nothing but the human. Active on reaching the small intestine. Proscolex everts or evagination and anchors to the intestinal wall. Now NX begins to proliferate, proglotted, and the bladder sooner or later gets detached and digested. Now, the parasite attains adulthood and possesses gravid proglottids ready for the apolysis. Clear? This is what the life cycle continues. Now look at this. See, what is the primary host? Here, man is the primary host and pig is the secondary host. See, in the pig intestine, the encysted hexagon will be present and the free oncosphere in the blood circulation. It has been released to the, after destroying all the membranes, oncosphere will be released and the blood circulation in this oncosphere will be circulate in the blood of the secondary host, that is pig. Now, the pig has been eaten. Cysticirce will be present in the further development. After that, this pig has been eaten by the human beings. Now the cysticircus becomes active. After getting active, it undergoes a further life cycle. What are all the things we have seen in the theory? This is the same thing will be explained in the picture. So you can find that. What are the parasitic infections seen in the Taenia solium? And we're going to solve some of the questions. Parasitic adaptations is tegument. External body covering or the tegument, it is freely permeable to water and nutrients but protects against the digestion by the host digestive juices. We have studied some of the hexacanth membrane will be lost only by the alkaline juices, but the hexacanth is not lost. How it is become possible means because of the body covering tegument. This is the structure which protects against the indigestion by the host alkaline digestive juices. No. Internal osmotic pressure is higher than that of the surrounding host fluid or tissue and pH tolerance is high. Adult as well as the larva lack cilia and other organs of locomotion. Everything I have explained here, right? This is what happening in the humans. No, see, every maturation will be happen only in the intermediate host, that is secondary host. What makes the difference means the cysticircus is inactive in the intermediate host, that is in the pig. Okay, cysticircus is inactive, but when it reaches the small intestine of the primary host, that is man, it becomes active. And after becomes active, the proscolex everts or evagination. Invagination happens in the intermediate host. Now, evagination happens in the primary host and anchors to the intestinal wall. Anchors to the intestinal wall means with the help of that hook, it attached with the intestinal wall. Now, neck begins proliferate, proglottids and the bladder sooner or later gets detached and digested. Now, after these morphological changes, this cysticircus will be developed into an adulthood. Okay, after losing some of the structures and gaining some of the structure, the cysticircus develops into an adult form. Now, it possesses the gravid proglottids ready for the apolysis, which means what? Further, they are going to give rise to many of the oncosphere in their fecal matter and the cycle will be continuing. Clear? See, this scolex with sucker spine serves as for the attachment with the epithelial lining of the host intestine. So the parasite may not be ejected from the intestine due to the parasitic contractions. Good. You have to understand here is scolex with the scolex, which is considered as the head. In the head part, what is happening? What is present there? Structures, suckers and the spines. It is for the attachment. With the help of these two structures, the parasite will attach with the host intestine. Alimentary canal is totally absent. The parasite absorbs readily available for the food for the host. As the digestive tract is completely absent, so it is going to take only the liquid food from the host so that the digestion process is not necessary. Okay, now what is structure helpful for the absorption from the host to the parasite? Means microvilli. Microvilli of the tegument increases the absorptive surface. No. See, the circulation, respiratory organs, sensory organs are absent. Nervous system is poorly developed, but it is having the nervous system, but it is primitive in nature, and respiration is anaerobic because the respiratory organ is absent. Why? Because the respiratory organs is absent means there There is no need of such structure because respiration followed by tinea solium is anaerobic. They doesn't need oxygen for their respiration. Okay. Now, this reproductive system is immensely developed. Okay, it has to multiply its population only because the respiratory system, the reproductive system is very well developed. Production of very large number of eggs, faces many challenges to survival of the species. Why it gives rise to that much number of eggs? Because some of the eggs will be destroyed. Because of the host is not present, some of the eggs will be destroyed. Only some will be undergoes a further life cycle. That is why this much number of eggs will be produced by those parasites to multiply their population. Resistant covering shell or capsule around the zygote. That is nothing but a capsule formation to protect the zygote or the egg cell during the unfavorable condition. Now, hermaphroditism and the proglotization ensures the self-fertilization. We have seen this. If self-fertilization not possible, cross-fertilization will be occurred. Now, the most important thing, what are all the diseases will be going to cause by this Teniosolium cystodiasis, and what are all the two different types of cystodiasis means teniasis condition and cysticercosis condition. The disease caused by the tapeworms. This is said to be as that cystodiasis. And what is the infection? Human being get infected by eating a raw and inadequately cooked measly pork. This is the only source of infection. And what does this pork contain? Cysticerce, which is an inactive form in the intestine of the pork. It becomes active when it reaches the human small intestine. Pathogenesis, what is going to fall victim to the adult tapeworm as well as to its cysticerce. Either the effects of the form will refers to the geniuses and those later is called as cysticerce. If the man is affected by this tapeworm directly, then it is said to be as what? Teniasis. If they are affected by the cysticerce means, then it is called as the cysticercosis. Okay, based on the things, we are going to study these two different conditions. TNSS and cysticercosis. It includes variety of symptoms, pain in the abdomen, nausea, anemia, increased appetite, indigestion, increased of eosinophils in cells. These are all the symptoms. And beside all these symptoms, nervous disorder of the type occurring in the epilepsy. Nervous disorder is just a nervous disorder which is caused by the TNSS. And the serious disorder is caused by the toxins. Serious disorder caused by the toxins, and who is going to produce this type of this toxin means none other than this parasite, and not as believed earlier by its continued drain on the host digested food. Hooks and suckers may cause the mechanical irritation in the intestine, may initiate reverse peristalsis leading to the auto-infection. And usually, a single tapeworm is found to parasitize a host. Anyhow, they are going to give rise to disease to the humans. This is because the parasite of one tapeworm provides a kind of immunity or pre-immunization to the host against fresh infection. Due to the what is peristalsis means the movement, only then it reaches the food reaches the stomach. Reverse peristalsis means again the infection or the parasite which is present in the small intestine, again they are going to reach the stomach. Once again the infection will be start. That is why it is also said that man is also act as a secondary host for this tinea solium. Now, cysticercosis, it is more dangerous than the teniasis. Encystment of the bladder may take place in the host voluntary muscles, cardiac muscles, and even in some more delicate vital organs like liver, eyes, and the brain. Removal of cysticercosis from these delicate tissues is very, very difficult. Okay, we can't imagine that much difficult it is. Cysticercosis of the brain results in several degenerative changes and necrosis in the brain, and the patient shows epileptic behavior, which means what? Nervous disorder. And what are all the treatments means? Anti-helminth drugs such as camoquine, carbon tetrachloride, alcorazine of the male aspidium. It is also said to be as the fern, quinacrine, antiphon, dichlorofen. These are all the drugs which are against the helminth parasite. But the most satisfactory compound for the human infection is yomesan. This is also a drug. Okay, that's all about the tinea solium. Any doubts in this? If you have any doubts, you can ask me, or let's move to the questions. Any doubts? If you have no doubts, you can answer for this question. The smallest tapeworm is, once again, because of time, I'll say you the important questions so that you can answer for those questions, only those questions. In tinea, cross-fertilization between different proglottids of the same individual occurs. This is called as the in tinea, cross-fertilization between different proglottids of the same individual. This condition is said to be as what? See what here they have given us cross-fertilization between the different proglottids. Cross-fertilization, different proglottids of the same individual. Then it is said to be as what? Very good. What is it? Self-fertilization. Now, hope you are getting very clear with the respect to the self-fertilization and the cross-fertilization. Bladder worm is found in human muscles, muscles of cow or pig, human feces, soil. Is it human feces? What we have studied here? Where is the bladder worm or the cysticercus seen in here in human? Where is the bladder worm? Here. In the human, it is not in the fecal matter. Clear? Then what is the answer? Bladder worm is found in. What is the answer? One minute. Bladder worm is found in muscles of cow or the pig. Okay, where it is present, we know that it is reaches the striped muscles. Okay, striped muscles of tongue, liver, lungs, like that. Striped muscles. Okay, so striped muscles of cow or pig. Bladder worm, which is in the inactive form in the pig, in the striped muscles. Clear? Only then it reaches the intestine of the humans. Where it is intestine of the humans. It is not present in either of these two. It is not present in the fecal matter. It is not present there. In the muscles of cow or pig, initially in the bladder worm. Okay, fine. Cysticercosis is a disease caused by the accidental infection of. Cysticercosis is a disease caused by accidental infection of. First option, hexacanth of tinea. Second option, bladder worm of tinea. And the third option is ancestor cercaria of schistosoma. And the fourth one is X of enterobius. What is it? Cysticercosis is a disease caused by accidental infection of hexacanth of Taenia, bladderworm of Taenia, encysted cercaria of Cystosoma, eggs of Enterobius. There is no connection between Enterobius in this question and Cystosomia to this question. Whether it is hexacanth or Taenia, bladderworm of Taenia. Just now I have explained. Cysticercosis is caused by what? Cysticercus, it is also called as the bladder worm. Okay, so obviously this is bladder worm is the one going to cause this diseases, which is nothing but cysticercosis. So the answer is bladder or worm of the Taenia. Okay, don't get confused. This is a simple question. Okay. Secondary host of Taenia is secondary host of Taenia is secondary host of Taenia is pig. And what is the primary host of Taenia? Primary host of Taenia is man. And Taenia lacks of digestive system, reproductive system, excretory system, all of these. Very good. Taenia lacks of digestive system. And larval forms of Taenia. Larval forms of Taenia, oncosphere, hexacanth, cysticercus, all. Very good. Larval forms of Taenia consist of all these structures: oncosphere, hexacanth, and cysticercus. Okay, fine. In which forms Taenia pass through the fecal matter of man? In which forms Taenia passes through the fecal matter of man? Oncosphere, hexacanth, cysticercus, young tapeworm. I have explained you, right? Very good. Oncosphere. No cysticercus. See, not a cysticercus. Lots of oncosphere will be ingested by the pig. Okay, lots of oncosphere will be ingested by the pig and reaches the and cause infection to the pig. Only then the embryophore and the basement membrane will be lost in the stomach of the pig, and the hexacanth together the hexacanth membrane will be reaches the small intestine, and again the hexacanth membrane will be lost in the small intestine by the alkaline juices. Now this hexacanth will be setting free and reaches the further organs. Okay, oncosphere, you have to say oncosphere. Again, pig acquired infection of Taenia by ingestion of. Pig acquire infection of Taenia by infection or ingestion of. Why are you getting confused? See, why are you saying cysticercus? Cysticercus will be developed only in the pig, but the pig acquire infection from the oncosphere. See, just remember, this is the fecal matter of the human. Okay, lots of oncosphere will be present in the fecal matter of the human. Again, it will be intake by the pig. What is getting inside the pig? Oncosphere will be getting inside the pig. In the pig, it is going to convert the cysticercus. So the answer will be the oncosphere. Clear? Now, Taenia saginata is commonly known as Taenia saginata is commonly known as Taenia saginata is commonly known as pork tapeworm, beef tapeworm, human tapeworm, none of them. Is it beef tapeworm? Ashanti Karnya, you're saying pork tapeworm. Then what about Taenia solium? Taenia solium is said to be as a pork tapeworm. So human saginata, we know that it is the tapeworm, but what tapeworm it is? Human tapeworm is the Taenia solium. I have explained when I teach you, Taenia saginata is also the tapeworm which going to affect the human directly. Okay, human tapeworm is Taenia saginata. Okay, pork tapeworm is said to be as Taenia solium. Knowing the common name is also important thing. Okay. Okay. Any doubts? So what's your duty is you have to solve all these questions. Because of time, I couldn't discuss all the questions. I'll discuss only the important questions. What is your duty is you have to solve all these questions in tomorrow's class. I'm going to ask you once again. Okay. Any doubts? Okay, fine. Thank you. Tomorrow, same time. Okay. You can leave the class now.